So now we come back to the lecture. As it was explained, I have made my lecture on indoor farming in order to be in phase, okay, with Professor Smith's lecture. So I come back again on this particular chapter concerning multi-omics has tools that can guide the discoveries of plant-specialized metabolisms. I want to explain you different things, and not in details. The idea is to give you an overview of what it is possible to do, and that it is what we do at this moment, but we don't know how it will be evolved, okay, during the next years. So you have now well understand that elucidating plant-specialized biosynthetic pathway has always constituted a laborious task. Okay, it's difficult to establish, firstly, the sequence of the chemical modification of a precursor to a product, and even if we think about different ways that lead from one to the end product, we need after to find the endogenous enzymes that are responsible of each step of this metabolic pathway. This is the main important task, and it is particularly important when we have some metabolites that are considered of high pharmaceutical values, because we need to produce them obviously in a high quantity, but not only pharmaceutical, in fact. So there was some very classical strategy. We can share together quickly what type of strategy was that are also currently done now. They have proven their effectiveness because this was the only way we had to identify several enzymes involved in metabolic pathway. But what it is true is that this methodology was quite long to identify at least one gene and to be sure that this enzyme encoded by this gene is really involved in the metabolic pathway. It could take about several years, depend on the difficulty to have access to this information, and also mainly if this enzyme be part of one of the biggest families of enzyme, and it is always the question. Now you know that. So with the emerging science, particularly omic-based science, we have seen a new process, a new way that had considerably facilitate and also accelerate the identification of these particular tools, such as enzymes that are involved in metabolic pathway. This is in particular important for obviously complex metabolic pathway, but also to have a better overview of how these pathways are cross-linking together, as we have now seen when I have present you the metabolic pathway. But you will see that if it is now a very nice set of tools, okay, it's always difficult to find the good genes. And what it is interesting is that as we have now considerably increased the number of genes known to be part of one or another metabolic pathway, we have now even enough material to engineer supplies, okay, in the cells, in the plant cells, but also in another type of systems, what it is named cell factories. So what it is interesting here to see is that in fact we have prepared all the new area of metabolite production, that it is based only on synthetic biology. So it will be interesting to see how these tools, molecular tools, are completely used for these synthetic biology approaches. So omic science, you know that very well now. It is a science that concern the entire, okay, the entire molecule of DNA in case of genomics, mRNA in case of transcriptomic, and proteins in terms of proteome. What we have already spoke about is the complexity of metabolite product in one cells or in one organism, that is the metabolome. To these 3 or 4 omic science, we will go in details for them. There is also other type of omic science that concern different subclasses of protein, for example, but also there is an epigenetic, epigenetic genome. I don't know how to pronounce that. There is also some fluxomic. I mean, it is someone discussed this some days ago. So you see that there is not only 4 or 5 omic science. You can apply this idea of science omic to several type of material, in fact. So it means that to have an idea of this, you need to be completely clear about the natural process from a gene to the enzymes, okay, that occur in each cells. What it is also true is that genomics, metabolomics, proteomics, and transcriptomics, they are the main omic science that we are using in our workflow, have not exactly the same level. For example, genomics, you know that it is the study of the organism genomes, the entire organism genomes. It is a raw material that it is present in all cells, whatever the type of tissue, whatever the type of the cell, the age of the cell. So, in fact, genomics can reflect all the genome in any samples you can have from a plant. So that's very interesting because, in fact, whatever the condition in which the plant is, if it is in light or at dark, under stress or no stress, the genome is always the same. So it is the base of the genetic knowledge of one of our organisms. It can be also a reference of the maximum information you can find in this genome. And in fact, we will see that genomic plants have made really significant progress in a recent year with new method of sequencing that have also permit to have access to complex and big genomes, and in a very short time and in a very low cost. So this is now possible to sequence quite easily and quite rapidly different type of genomes. Here, just precision, genomics and genetics are not exactly the same. I saw that for my own students, they make confusion. They have confusion on this sometimes. So this is why I have put this information. So genomics, it's the study of an organism's complete set of genetic information, while genetics is more dedicated to the study of heredity. So how some gene are some allele of gene are transmitted from parents to child, for example. So it can be interesting because genome is a complex genetic information, while genetics is focused on specific genes, and specific sequence of DNA. And if we take a look of what happens with metabolomics, proteomics, or transcriptomics, it's completely different. Because even if it concerns for example, for proteomic, all the proteins that are produced in an organism, okay, it is quite different because depend of the cells, depend of the organelle, depend of the tissue, and also depend the time and the condition in which the plant is or the tissue is. So it's like a snapshot of one particular moment. So it's not also complete. It is not like a genomic science. That it's completely independent of this specific condition in which is your raw material, so the plant in this case. And it is exactly the same for transcriptomic and for metabolomics. So it means that before starting to make some proteome, transcriptome, or metabolome, you need to be exactly conscious of what is the situation of your plant. And this is interesting because by this way, we can propose to put the plant in different type of condition. And by this way, also you can compare how the proteomes, for example, or transcriptome or metabolome, can be conserved or can be different. And this is this different expression of transcript, metabolite, or also protein that can indicate us what are the specific genes that are involved in a specific condition, for example. So these three other, yes, j'ai peut-être fait une bêtise, sorry. So these three types of omic science are very useful for deciphering, in fact, the importance of a specific type of gene involved in the answer of a particular condition. So what I propose you, now if we are agree with the idea of the general concept of omic science, is that what are the workflow that have been developed using this type of omic science for discovering new genes and also new branch of a metabolic pathway. So perhaps before entering in this world, I can also check with you the classical strategies that have been already done and that are currently tried, in particular when we are in presence of a plant for which we have no genomic data and very few transcriptomic or different omic data. And sometimes it's quite different to obtain easily them. So we need to come back to traditional strategy if we want to discover different things. One of the first strategy that have been developed is a classical one, is based directly on purification of a specialized metabolic enzymes for gene identification. Means that the idea was we have some plant or cell culture or plant raw materials, okay? We can, after a release of this plant cell culture, we can make some chromatography in order to separate all the protein in subfraction, and each pool each fraction can be assayed according to a specific enzyme activity, because you are looking for example, a P450 protein enzymes. So you have established an in vitro test for showing this type of activity. So you can test a fraction of your purification process to see if the activity you are looking for are present or not in the fraction. If the activity is present in several fractions or subfractions, you can pool them, and after you can separate all the protein by a classical polyacrylamide gel electrophoresis strategy, okay? And by this way, you can separate all the protein. You can extract the band of the corresponding protein, and if you are not in if you have a native page, okay, you can also try to test directly the activity of the protein, but it is quite difficult to have a pure protein in this case. This is why we use denature. Okay. Okay. I have some difficulties this morning. I don't know why. Sorry. So we can extract the protein that is not active. So by this way, we can sequence the protein, okay, the amino acid protein. We can deduce the synthetic, okay, oligonucleotide corresponding to the amino sequence of the protein. And by this way, we can use this sequence as a probe to screen a specific library named cDNA library in order to isolate which can be the clone that can express specifically this protein. Just a slide to, I don't know if you know how to produce complementary DNA libraries. So just a slide to be sure, okay, that we have exactly the same information. You know that from the DNA, there is a transcription step leading to specific mature mRNA. I go fast, okay. This is a single strand nucleotide sequence. And what it is interesting is that we have now several enzymatic tools, such as reverse transcriptase, that are enzymes that are able to complement, okay, the mono strain of mRNA in order to have a double strand like a normal DNA molecule. This is interesting because this mRNA have been completely avoided from the intron. Okay, only exons have been conserved, so the entire sequence corresponds to the coding sequence, okay, of the gene. So by this way, you may have directly the sequence of the mRNA that it is directly translated in a protein. And what it is interesting, if you do that with all the transcriptome, so you have to isolate the transcriptome, but you can make a sort of complementary DNA library by including all of these two string bands in several plasmids that are after insert into Escherichia coli, and you can conserve all these fragments of cDNA separately, okay, in each E. coli cells, and all the genes are represented at least in one type of cell. Sometimes it can be more, depending on the number of your message, but in fact you have at least one enzyme, bacteria that it is able to have at least one of the inserts. So this is what we call a complementary DNA library. What it is interesting with this type of library compared to a genomic one, that could be also done by the same way, is that in fact you may have representation of how the mRNA was abundant at the beginning of this process. Okay, I will be more clear. For example, if you take some genomes, you cut the genome, okay, by different way. Enzymatic one can be, but you have, okay, library that represents the number of the gene you have in the genome. But when you have a transcriptome, as I said before, it's at a particular moment, means that not all the genes are present in the transcriptome. So the cDNA library is also not complete as a genomic library could be. And also what happens when you have a sort of small set of mRNA, they are not expressed at the same level, depend of stimulation of their expression. So in a cDNA library, you may have more cells, prokaryotic cells that have exactly the same cDNA in their plasmid, while some are represented only by 1 or 2 cells. This is a reflect of the different amount of expressed gene in the cell. So this is also interesting to have an idea of which are the more transcript genes instead of what the genes that are more less expressed. This is the case, for example, for gene encoding enzyme of metabolic pathway. They are not overexpressed as other type of gene. So when you have this type of tool, it can be the base for screening, okay, when you know how to screen. What it is exactly here, what I have tried to explain to you. If you have these probes, nucleotide probes, you can screen this cDNA library, and also you can see which clone of Escherichia coli may have exactly the sequence that corresponds to the gene encoding the protein you have isolated at the beginning. And by this way, you have the cDNA, the complete cDNA that encodes directly the protein you are looking for. This was a way to identify several specialized metabolic enzymes, okay, for several years. It can be also continue to be done by this way, but it is a very long and laborious, as I said before, works. A second way that it is also based on the use of a transcriptome, okay, is to have an idea of homology-based cloning. Means that when you have different medicinal plants, okay, you can extract the transcriptome, and after, by several techniques, you can sequence all the transcriptomes. So you may have this information at this moment. On the other side, you have an idea of what type are the different modification of a precursor to lead to the final molecule. And you know that you have several enzymes that catalyze each step of this, for example, portion, okay, of metabolic pathway. You can imagine here, you know the first one, second and third one, and the last one, but you don't know this enzyme. You are looking for this particular step of the metabolic pathway. What you can do? Chemists can help you because they said that between this and this, there is a specific chemical modification, and now you know that to a chemical modification, you can link a specific enzyme activity. So you may have an idea of what is the family of enzyme that you are looking for, one specific enzymes, okay, that be part of this family that could catalyze this specific step. So what is interesting is that in the literature, you have an idea of what type of enzyme here. You can have all the sequence of this type of enzyme, not in the plant you are interested in, but in other plants, okay? So all of this sequence can be compared, and by this way, you can find several short sequences that are well conserved, okay, in all of these genes that encode this type of enzymes. So these short sequences are very interesting because they can be used to design a specific oligonucleotide that can be used to make some PCR, polychain reaction, in order to amplify in your own cDNA library that come from transcriptome analysis, genes that have this type of conserved sequence in the enzyme family, okay, that correspond to this sequence. So by this way, you can amplify one or perhaps sometimes 2 or 3, okay, bands that share, okay, this type of conserved small sequence. after that, you can extract all of these amplified gene fragments, and we can use this type of fragment as a probes, like it was in the previous strategy, to screen a cDNA library, okay, that you have produced also depending the plants, the moment, and the situation of stimulation. In particular, we try to develop here some cDNA library of plants that are in a high production of the metabolite. We want to know the metabolic pathway, okay? And this is because we thought that it is a way to have expressed all the gene encoding the metabolic pathway at this moment. And by this way, you can have also, after this screening, some clone that have fragment of the gene, and by this way you can rebuild the entire sequence of the genes. That could be done, but also you can have the opportunity to express this gene and to have what we call the recombinant protein, so the corresponding protein, and after you can validate the activity of this protein in vitro in order to be sure that it is the enzyme that is able to convert this product in this product, for example. The limitation of these systems, it's a quite efficient, long but quite efficient, but the limitation of this system is when you want to validate the function by enzyme activity, you need to have these intermediates. So sometimes it's quite difficult to have the metabolite intermediates of the metabolic pathway. And even if you discuss with chemists and if you ask them, can you synthesize for me, for example, a part of this intermediate? Is it possible? But sometimes these intermediates are very unstable. So it became difficult to have efficient biological assay, activity assay, okay, to be sure of this activity. So we have now some other strategy to confirm, to validate the function of the gene. I will show you, perhaps not today, tomorrow. Okay, so taking this in mind. So this is also, and it is still one of the strategies that can be used, that use in particular transcriptomic based omic, just to have a material for make the good screening. Is it clear for all of you? I don't know if you have the molecular background, okay, to understand all of this type of strategy. So don't hesitate to stop me, to ask me in case of. Okay. Also, we will take time to discuss about it after. One of also interesting things that have been particularly developed for metabolites that have some color property, for carotenoid, for example, in general, is to screen what we call an expression bank. What is an expression bank? An idea? We have spoke about cDNA library or bank. This is the same. What is an expression bank? I don't hear you, sorry. Quite. In fact, it is when you have a cDNA, a full, okay, length genes from plant cDNA, means that you can insert this cDNA that encode for a specific protein in a specific plasmid that it is dedicated to translate, to transcript and after translate, okay, this particular cDNA. Means that instead of having just a prokaryote cell inside the plasmid with a cDNA sequence, this particular cells is also able to express this cDNA and accumulate the corresponding protein also in the cells. This is what we call expression bank.
Similar to plasmids. So are they similar to plasmids?
It is similar to?
Are similar to plasmids vectors that carry?
Yes, depends on the vector. The vector is not exactly the same. You have all the sequence needed to express the cDNA that have been included in the plasmid. Okay, but there is now a nice collection, okay, of this type of expressed vector that lead to production of corresponding protein that are recombinant proteins too. Is it okay for you? So means that I just make here. If you have this type of libraries, instead of having only the fragment of cDNA, you have the plasmid, but also all the protein that, okay, that are directly translate from this cDNA information. So you have directly what you are looking for. But how to screen in this library, okay, the correct enzymes you are waiting for. But in fact, it is exactly what I say to you. You have all the E. coli cells, for example, E. coli cells, and you have just to give to these cells precursor of this enzyme activity. And what it was interesting for example, terpenoids or specifically carotenoids, is that the products that are produced by the enzymes are a colored molecule. So after, when you put all your bacteria on a medium and you wait to have some independent colonies that correspond, each colony will correspond to one particular E. coli cells at the beginning. And some of them become, for example here, yellow. Means that the enzyme that are able to express is able to convert the precursor you have given to the cells, okay, in the final product you are waiting for. So means that these enzymes that are expressed in this colony are probably enzymes involved in the metabolic pathway of carotenoids. So you can have only, you can have to isolate after this clone to extract the protein from these specific colonies, okay? And you have directly, okay, the enzymes you can make after in vitro assay in order to confirm completely the function of these enzymes. It is also a way to screen several types of substrates in order to see if this enzyme could be also involved in other type of substrate modification. So you can do a lot of things with that. And after that, you can conserve preciously the clone expressing this molecule in order to have, when you want, and if you need, the protein, you have just to cultivate these particular clones and also to extract after the corresponding enzymes. So this is very nice process to obtain directly the protein and the gene after, but it can be limited by the screening you have to develop for this specific bank, okay, expression bank. If it is, for example, not a colored protein, metabolite, you need to find other type of screening systems. It can be to include in the medium, for example, at what moment, I don't know, a specific molecule that can be converted indirectly, okay, and in a molecule you can easily find. So it can be limited depending of the type of activity you are looking for. But it is another type of strategy. So, and the last one that it is not, it is already developed now is to make some correlation between two types of data, in particular, gene expression, yes, okay, and also metabolite production. Here, what it is interesting is to have two differentiated condition of production and condition of non-production. You need to have very contrasted situation in order to be sure to have a specific element that could be linked to production of metabolite you are looking for. So by this way, you can do two types of analysis. The first analysis is to isolate the transcriptome, okay, of the cells that produce the natural product and the transcriptome of the cell that not produce the natural product. And after, you make what we say a differential display, okay, of the expression of the cDNA in both of example. You can eliminate all the common expressed gene, and you can after be focused only on the gene that are specifically expressed in one condition or in the second condition, because not only those that are expressed in the condition making, okay, the metabolite are really interesting, but also those that are downregulated, for example, in another situation. This is a way to access not only to one genes, but to several genes of a same, okay, metabolic pathway. So it is more interesting for this region. And what it is also interesting is that if you have also isolate all the small molecules, means you have done some metabolomic study, okay, you can monitor, in fact, the gene expression and the metabolite expression to make what we call the gene to metabolic network. And by this way, you can correlate. It is some statistical analysis of your data. You can correlate the presence of several metabolites with the expression of several genes. And by this way, you can have an idea of if the gene could be involved in the metabolic pathway. It is less precise than the methodology I showed you before, for which you targeted a specific event in the metabolic pathway. Here it's a way to have more an idea of what type of gene could be involved in those metabolic pathway. Okay? I show you like that. I don't know if you understand all I am talking to you. Is it okay for everybody? Yeah. Because with this I can't hear you, because I have a noise with
Hello. So, ma'am, you just said that related to this gene expression for metabolite correlation, that several metabolites of several genes are expressed.
Yeah.
So, further you can identify the genes of its role in the pathway. Can we also use this to identify the role of the gene in another pathway?
No.
No.
No. Yes, after, when you have it, okay, you can have the corresponding protein, and after you can test what you want with this protein. For example, you can test several types of substrates, including substrate of the metabolic pathway you are working on, but you can test also some other type of metabolites in order to see if this enzyme may have a more large set of natural substrate. That's true. But in general, they are more dedicated to a specific type of metabolic pathway. Not so specific on the branch, but at different steps sometimes of the metabolic pathway. I showed you last day that, for example, you can have one enzyme that acts at a first point, after it is another one, and at the third point it is the same enzyme as the first one. So in fact, by this way, if you have the recombinant proteins, you can test several things only if you have the intermediate substrates. This is always a limitation. If you haven't all the the intermediate metabolite substrate, it became difficult to do this type of analysis. Okay. So, what happens now? Because all of these strategies I show you are classical strategies. So now what we can observe, it's a complex figure, I know, but it is a nice one. This is why I have put, and I propose you to discuss it a little bit. In fact, what happens is that we have a lot of data for a long time, transcriptome, genomes, and also proteome, metabolome. This was quite not easy, but it was for a long time we can do some. But what we needed, it was to have some specific algorithms that could be able to analyze such amount of data in order to try to correlate them the one to the other. And what we have observed is that from 1997, I think it is here, we have started to develop, we, scientists, not me, I am not expert in this area, but scientists have developed some computational genome mining tools that lead to identify quickly, okay, some metabolic pathway and some genes involved in this metabolic pathway. I like this picture. It is a picture that I have taken from a very recent review, particularly that is dedicated to what we call gene clusters. And we discuss this with you after. But here you have different strategy. Before, for example, metabolomic was used alone, okay, to try to decipher some of specific gene missing in a metabolic pathway. Or transcriptomic, it is in green, or genomics, okay, here it was in orange. But what you can observe is that when, with the time, until quite now, we have combined not only one, but two, sometimes three or four, okay, omics science, in order to be more precise, in order to discover directly the genes that are missing in several parts of a metabolic pathway, for which sometimes it was 20 years that we are looking for the genes. For example, it is by one of these type of approaches that in my lab and with one of our collaborators, we have found the genes that was looking for 16 years. So it was not so easy to find, okay? And this is what it is interesting. You can mix, for example, genomics with metabolomics approaches. Here are some of the genes that have been identified in this specific type of plants, Japonia, and also you can combine genetics with transcriptomics. So means that you mix genomes and also transcriptome. But what we observe here is that the last year, the more efficient strategy that are used to discover specific new genes combine metabolomics with transcriptomics. So means that in fact, now we can have some integrated, okay, study of this omic data, and this could be faster to identify, in this case, not only one, but sometimes several genes that are involved in a specific metabolic pathway. So this is interesting to see that, because in fact, all of these genes that have been, genes or set of genes, okay, that have been identified during all this time, now serve as a reference for finding new other genes by the same methodology.